Welcome back, grade 12. So we will be continuing with Unit 2, Meiosis, and for this lesson we are focusing on the process of meiosis. So meiosis forms gametes, which are the sex cells in animals, or spores in plants. And during this process, four daughter cells are produced, and these daughter cells are not identical to each other or to the mother cell, as you can see here. Okay. The chromosome number is halved from diploid 2n to haploid n. Okay, this is called reduction division. So the chromosome number is reduced. Okay, so as you can see here you have 4 chromatids and there are only 2 chromatids. Okay, so the chromosome number is reduced. Okay, so if the gametes were diploid 2n, the zygote would have twice the normal chromosome number, and this is why meiosis is so important, because if meiosis doesn't reduce the chromosome number back to the haploid, it would just keep increasing and increasing and increasing with each subsequent generation. So you would have a mom and dad with 2n and 2n, children with 4n, they would have children with 8n, they would have children with 16n. So meiosis prevents doubling by halving the chromosome number. Thus, when gametes n fuse, the diploid number 2n is restored. Meiosis is a type of cell division that produces 4 different haploid daughter cells, as we can see here. Right. So the process of meiosis. Meiosis is a continuous process that is divided into 2 stages. The first meiotic division, or meiosis 1, and then the second meiotic division, or what we call meiosis 2. During the first meiotic division, chromosome number is halved and the genetic material is exchanged to produce variation. The second meiotic division is almost exactly the same as mitosis. So where the sister chromatids separate from each other is during this process. And if you are a little unsure about mitosis, you are more than welcome to use this section to recap that as well. So both meiotic divisions have the same 4 phases as mitosis as well: prophase, metaphase, anaphase, telophase. So prophase, meaning at the beginning of the meiosis process; metaphase, when the chromosomes lie in the middle; anaphase, when they are on opposite sides, like antonym meaning opposite, so opposite sides; and telophase, when we have 2 separate or 4 separate cells that have to phone each other. Yes, telephone. Okay. So before meiosis occurs, interphase happens, and we know this as well from mitosis, during which DNA replicates and 2 copies are formed and the centrosomes duplicate. So let's start with meiosis 1, prophase 1. So in prophase 1, at the beginning, the chromatin network condenses and the threads shorten and thicken to become visible chromosomes. So then we move from something like this to something like this. Okay? They separate the separate chromatids cannot yet be seen, and the homologous chromosomes arrange in pairs and lie in contact with each other. So this is when we call them ambivalent. So like these 2 that are touching each other, they are ambivalent, okay? They are homologous chromosomes. They appear as a single chromosome with double thickness, okay? And then in the late stages of prophase, the homologous chromosomes become visible, and then it looks like what we see here. Okay. So now just to explain that overlap and bivalent situation to you. Overlapping occurs between 2 adjacent chromatids of the bivalent, and crossing over happens. So during crossing over, segments break off and are exchanged between the 2 chromatids, as you can see here, that section and that section, right? So they crossed and then a section broke off and switched out, okay? The point where they cross, it's called the chiasma. So this is where they cross and break off, is the chiasma. And then this brings in genetic variation due to the reshuffling or recombination of the genetic material in these chromosomes. Okay, so now there's variation. That chromosome is not like it was originally. There are some changes. Okay, so there's variation. Then your nucleolus and nuclear membrane will disappear, as you can see here. And then the centrosome splits and the centrioles move to opposite poles of the cell. So this was the centrosome that splits, and now they were able to move to the opposite sides of the cell. Okay. The spindle fibers form between the centrioles in order to form the spindle. Okay, so they form a spindle between these two centrioles. Okay. Then we get to metaphase 1. So the homologous pairs of chromosomes are positioned on the equator in a double row, like this. One chromosome on either side of the equator, and some spindle fibers attach to the centromeres of these chromosomes now. Okay, so the spindle fibers attach to them, and then again we have random arrangement, which brings in some more variation. This just basically means that in the cells, these 4 chromosomes arranged like this, but they could have also arranged with this one there and that one here and whatever. So then those daughter cells will look different. So again, this brings in variation. Then we move to anaphase 1, during which the spindle fibers contract and shorten, pulling the chromosomes towards the poles, as you can see here. The chromosomes of the homologous pairs are separated. So these two were homologous pairs and those two were homologous pairs, but they are now pulled away from each other. So they are separated. And one chromosome of the homologous pair moves towards the one pole and the other towards the other pole. So they are split up between the 2 poles. Okay, the 2 homologous or the homologous pair. Then cytokinesis will also begin. So the cytoplasm will begin to divide, as you can see here with this little indent. Okay. Then we move to telophase 1. Now during telophase 1, the chromosomes group at the poles, like you can see here. They form this little group, and the spindle fiber will disappear. You can see there's no spindle fiber here anymore. A nuclear membrane forms around each of the 2 groups of chromosomes, and a nucleolus forms in each. So there you can see you have your nuclear membrane that formed and your nucleolus. Okay. Then cytokinesis occurs, and each of the 2 daughter cells has 1 chromosome from the original homologous pair. Okay. So remember we started with 2 homologous pairs, and now we have 2 separate chromosomes from each of those pairs. So we had 4 chromosomes in the original one, and now we have 2 in this one. Okay, let's just show you that. There we go. There you can see we have 4 chromosomes. So each cell has 1 of these from each pair. Okay. Then the chromosomes in the daughter cell have the chromatids joined by a centromere. Again, we have the centromere that just joins the chromatids of the chromosome. Okay, so this is the end of meiosis 1. Now we move on to meiosis 2, which is your prophase. Okay, prophase 2 is at the beginning of meiosis 2. So again, each chromosome has 2 recombinant chromatids that exchange genetic material, and they are joined by a centromere. So this is where we ended off with the previous cells. Those are those 2 cells. So they now have the recombinant DNA because of that exchange that took place earlier. And yeah, okay, so there are no homologous pairs, remember, because those homologous pairs split up in meiosis 1, and a nucleolus and nuclear membrane now starts to disappear, and the spindle will begin to form from the centrioles at the opposite poles joined by the spindle fibers. Okay, chromosomes in each of the cells have 2 chromatids, which is joined by a centromere, just like in the previous slide. Okay, Then the chromosomes move to the equator and align on the equator, but this time they are in a single row. Okay, as you can see there. And some spindle fibers attach to the centromere. So again, spindle fibers will attach to the centromere of these chromosomes. And random arrangement of chromosomes again introduces variation in the daughter cell, because in this cell, for example, they are aligned like this, but this one could be switched around in another cell. So, so that's again random variation that happens. Then we move on to anaphase 2. So the centromere of each chromosome divides. Two sister chromatids separate and move to opposite poles. So the centromere that was connecting the chromosome in the previous slide now divides, and then because of that, the two chromatids are able to separate and move to the separate poles. So the centromere is pulled by the spindle fiber. So these fibers pull the centromere towards there. Remember, the centromere is the little circle in the middle that connects the two chromatids of the chromosome. Okay, and then cytokinesis begins to separate the cytoplasm, again visible by this little indent. Then we get to telophase 2. So during telophase 2, the chromatids group at the poles and are known as unreplicated chromosomes or recombinant chromatids. So now you don't have chromosomes anymore, because those chromosomes split up into their chromatids. One chromatid to this side, one chromatid to the other side, right? And they are recombinant because they had genetic variation and crossover that took place earlier. So your recombinant chromatids now move to the separate poles, and the spindle fibers will disappear, as you can see here. A nuclear membrane forms around each of the two groups of unreplicated chromosomes, and a nucleolus forms in each. Like you can see here, your nuclear membrane, and then a nucleolus that forms in each. Cytokinesis will occur, like we can see with the indent, and after cytokinesis is completed, 4 cells are formed, each with the haploid chromosome number. Okay? So they are not diploid, they are haploid. And the daughter cells are genetically different due to the genetic exchange that happened earlier. The daughter cells develop into sperms in males and into eggs or ova in females. The 4 daughter cells have half the chromosome number and chromatids only. So again, at the beginning we had 4 chromosomes, right? Then at the end of meiosis 1, we had 2 chromosomes, and now we have 2 chromatids. Okay, and that is meiosis 1 and meiosis 2, which is your process of meiosis. Please make sure that you are able to draw this to explain the process. It is vital. And then I'll see you in the next lesson when we look at the purpose of meiosis, as well as its importance and what happens when the abnormal meiosis occurs.